Hello everyone. So today we are going to be doing our second lecture in the Intro to Genetics series. Today's lecture is going to be all about chromosomes. So we will talk about chromosome structure, how they are made, and then how they're maintained, and then what their function might be. So we will be doing a little bit of that today. We'll continue that discussion next lecture as well as we talk about how they are divided in mitosis and meiosis, and then we will talk about some rearrangements and some changes that can happen when something goes wrong during replication and what their repercussions might be. So let's go ahead and get started. As usual, I have the links to the textbooks that I'm using. Those are both open source textbooks, so you can easily access them in the description below. And I have added in the lecture information on the sections and chapters that I will be covering in today's lecture. And moving forward, I will have that information available for you in all the lectures that I post in this series as well. So, overview of what we're going to be talking about today. We're going to go back and kind of review what DNA is, what it's made of. We're going to talk about how DNA is packaged inside the nucleus in the form of chromatin and chromosomes. We're going to talk about what karyotypes are, and then we're going to be looking at some polyploidy changes that happen when um, cells divide in a different way or when mutations happen. And then we are going to be finishing up with endoreduplication and gene balance at the end of this lecture. So to start off, we're going to be looking at DNA, chromatin, and chromosomes. So we looked at DNA last week and we looked at its basic double helix structure and what it is made of, which is that sugar phosphate backbone and the two antiparallel strands with your nucleotides inside them, the base pairs that are inside them, where you have the AT pair and the CG pair. Um, it is one of the largest human, molecule that you have inside your cells in the form of our DNA of the nucleic acids. The largest human chromosome that we have in our cells is 85 millimeter if it is stretched all the way. That's a really, really long size considering the cells are usually in microns in size. So it essentially has to be compacted thousands of fold, up to 10,000 fold to be inside that nucleus. So when it is fully compacted, it actually only takes 5 micrometer space during cell division. So that's a really tiny percentage of that full length. That kind of gives you an idea on how much compaction of DNA happens in our cells on a daily basis. Um, However, the ability of the DNA to be compacted so much allows for very easy transport in dividing cells because they can be easily kind of packaged up and divided when it's time for them to divide. Um, Now, just because they can be compacted doesn't mean they're always in that state. Different chromosomes, depending on the cell's needs and the cell function that it is performing, are going to have different levels of compaction. Even in the same chromosome, some parts of the chromosome are going to be more tightly compacted than others. Some are going to be really open because they want, you have genes in there that need to be at that moment transcribed and translated for the cell to do its function. Um, so depending on what stage of cell cycle you're at, depending on what the cell performs, what functions it performs, what its needs are, you will have different chromosomes in different levels of compaction. And that changes for cells on a daily basis, on hourly basis at some times. When you are looking at the chromosomes inside the nucleus, you will see these are just decondensed chromosomes. These are not fully compacted chromosomes. This is how they would be in a typical setting inside the nucleus. It's not just that these chromosomes are just kind of haphazardly thrown inside that nucleus. They have actually specific locations that they can be localized to. That is, pretty consistent. So we can see that through a technique called FISH, which is fluorescence in situ hybridization, where you take your nucleus and you are using fluorescent labels to, you take the cells actually in culture and you're using these fluorescence labels to hybridize two various chromosomes using different types of stains. Um, and then when you do that, usually we use 7 different fluorochromes. And when we use them in different combination, each chromosome essentially shows up in a slightly different color or pattern. And you can use that information to get an image like this one that you see down here. Um, and you can localize where each chromosome is hanging out inside that nucleus. So in this particular case, this is a fibroblast nucleus, and so you can see all the various chromosomes at different spaces inside this nucleus. And you see some of them are taking a lot more space than others, and that can partly be indicative of how active that chromosome is or how large that chromosome is in its normal setting. Oops, sorry. So looking at the DNA again, let's look at the various levels of compactions that happen over the course of a cell cycle or a cell's life cycle. So without anything attached to it, the DNA itself is just this long molecule, right? Depending on the size of the chromosome. And it just has those two sugar phosphate backbones and the base pairs. That's your plain DNA without naked DNA, without anything else on them. The first level of compaction requires these histones, these core histones that come together to make this complex, where the DNA wraps around these histones and creates these nucleosomal spaces. So the nucleosomes are composed of these histone molecules, 8 of them actually coming together to create a kind of a yo-yo looking structure that the DNA wraps around. And that's that first level of compaction. Now, when the entire chromosome has these nucleosomes with the DNA wrapped around them, you get this kind of bead on a string look. So this is essentially a strand of DNA with just the histones there and the DNA wrapped around them. So all these nucleosomes. Um, and what you will see is that the DNA is wrapped around each one of these about 2 times. And then there is a little bit of a linker DNA as an open space, and then the next complex is there, and then same idea. And if you were to calculate that distance, that's a consistent distance. It's always around 200 base pairs. It's like 188 base pairs. So about 200 base pairs from one histone to the next, or from one nucleosome to the next, rather. Then another histone, an H1 histone, gets added into this mixture and into this bead on the string look to further compact this molecule so that it is even more tightly coiled and linked. This creates this 30 millimeter fiber. And here you can see how that one is now beginning to look more like that coiled structure that we associate with a chromosome. Now, normally, this is the way that you're going to be finding these molecules inside the nucleus, where they are, some of them are going to be at this level of compaction. Some may be a little bit more uh, compacted using scaffolding proteins. But this essentially, this 30 nanometer fiber is using the scaffolding proteins to stay in place in that large nucleus that it is hanging out in its own location. And some of it is going to be more open, some of it is going to be more tightly packed. The more scaffolding proteins are present, the more tightly it can get packed as well. Now, here in this first panel, in this electron micrograph, you can see this 30 nanometer fiber. That's what you're looking at there, where you can actually see those H1 histones and the nucleosomes all coiled together to create that structure. Now, your interphase stage is where, the general cell cycle is happening, but it's no mitosis, no meiosis, no actual cell division is happening. This is where these cells are kind of gonna, the DNA molecules will hang out at that stage, is in this kind of nucleosomal, complex in this 30 nanometer fiber. But then during metaphase or during mitosis and meiosis, when there is active cell division happening, and you need to have your full chromosomes fully compacted, more scaffolding proteins come through, and these DNA molecules now become compacted in a very organized manner. So an interphase, if you see, it's kind of a loose organization. It's not a very, organized manner. It's just kind of loosely attached scaffolding proteins hanging out in that nucleus. But now they become in a very particular structure and a particular way, with the help of other linker proteins and scaffolding proteins that end up helping you create this chromosome structure that we know as our DNA chromosomes. So looking at those nucleosomes a little bit more first, so starting off again from the DNA, that first part of compaction comes with the help of these nucleosomes. So the nucleosomes, like I said, contain several histones coming together to create that complex, that yo-yo looking complex that the DNA wraps around. And there are 8 total histones that are going to come together to create this. So this is the quaternary structure of that, of this nucleosome, right? You have 2 histone 2As, histone 2, histone 2 of each. So histone 2A, 2B, histone 3, and histone 4 that are going to come together to create this nucleosome. And then one of them, histone 3, H3, has kind of a little N-terminal tail that is used as a way to clip and hold the DNA together. And it kind of sticks out towards the outside from that internal yo-yo round structure. So looking at that, the DNA is going to get wrapped around the structure twice, and then it will have a little linker, and then you will have another one of these. So if you were just to take that coiled structure, just this structure that is the amount of DNA that is coiled around that octamer, that histone octamer, you would get 147 nucleotide pairs in that structure. And then once we combine that with that linking in between linker DNA, you would get approximately 188 to 200 base pair. So that is a very constant measurement all throughout that long chain of DNA. We know this because we've used nucleases like DNase to digest those linker DNA and just take that nucleosome core particle and release the amount of DNA that's on there using high salt concentration solutions. And we can look at the fragment length and know how big it is, and it's very consistent. Um, so nucleosomes essentially contain the DNA wrapped around the the protein core of it's an histone octamer containing 4 different types of histones coming together to make that molecule. Now, chromatin packaging inside the nucleus, like I said, varies depending on the cell cycle stage and the need of the cells. And the chromatin is what we term chromatin is this itself, the chromosomes and the proteins together that are compacting together, that are making this complex together that eventually make up the chromosomes. That's what we call chromatin. Um, there are 2 different types of chromatin depending on how it is compacted. The first is euchromatin, which is the more loosely packed chromatin. And this is where you tend to have the genes that are being actively transcribed at that time, or that are like housekeeping genes that are always going to be present, or genes that are related to the needs of the cell at that given time, at that part of the cell cycle. And then you have heterochromatin that is usually related to the sequences that are not transcribed nearly as often. It doesn't mean they're never transcribed. Much of it is still going to be transcribed at some point during cell cycle, or some of it at least, but they are not used nearly as often, and they are going to be more tightly packed. And in heterochromatin, you will have areas that are super tightly packed, and others that are not going to be as tightly packed. But this tightly, densely compacted DNA is typically rich in a lot of repetitive sequences that are not usually, like I said, transcribed or transcribed as often. And then it will also contain these very short, highly repetitive sequences that we call satellite DNA. That is almost never transcribed for any kind of protein needs. So those two terms you would want to keep in mind. Now, next, we're going to be looking at now the final chromosome structure that you get once all the levels of compaction are complete. So your chromosomes are basically these rod-shaped filamentous bodies that are present in the nucleus. They only become visible during active cell division. So all the rest of your cell cycle, they are going to be present in the form, the chromosomes are going to be present in the form of chromatin, which is your DNA and protein complex, but they are not in their perfect little compacted, fully compacted shape of chromosome. They are the basic carriers of the genes of and your basic unit of hereditary. They are not going to be active, are not going to be visible in an active nucleus, partly because to their high water content, but also because basically they're not compacted fully, right? They are more loosely compacted. They have areas that are being actively transcribed, so they're all unraveled up in that area. And so you're not going to be able to see them the same way as you would in active cell cycle. The name of a chromosome, the term chromosome, comes from chromo, which means color, and soma, which means body. And that's because initially, when they were looked at under the microscope, they noticed these bodies that would pick up certain type of basic dyes and change color and give you like some dense, some banding patterns or some different colors. And because of their affinity for these basic dyes, they ended up calling these molecules chromosomes. So let's look at some morphological features of a typical chromosome. A chromosome is typically going to look like they have two arms or two sets of arms. The shorter of the two arms is going to be termed your P arm. It doesn't matter whether it's on the top or the bottom, but the shorter arm is going to be the P arm, and the longer arm is going to be your Q arm. They also have other distinctive features that are important for their structural integrity and their function. One of them is the centromere, which is an area of the chromosome where the two sister chromatids are connected. And the two sister chromatids are basically when you replicate your DNA, they are the exact copies of each other. Uh, so those two sister chromatids, the region where they're connected is termed as centromere. That doesn't have to be in the center. It can be off center to one side or the other, hence the short arm and the long arm. They're made up of repetitive sequences and they combine together with proteins to create that centromere structure that then binds to microtubules during cell division. And we'll learn all about that later on in the um, mitosis meiosis lecture as well. But I've also talked about them before during my molecular and cellular biology series, and I'm going to link those videos down below when I've talked about microtubules and cell division before. So you can look at that for more details on that structure and what proteins are combining together to create those centromeres. The centromere is what we term as our primary constriction in the chromosome. You can also have secondary constrictions, usually towards one side. And those secondary constrictions will have a little bit of DNA then left behind on the top, and those are called satellite DNA structures. Another important part of chromosomes are the telomeres. Telomeres are repetitive TTA-GGG DNA sequences, so that exact six-base pair repeated many thousands of times at the end of the chromosomes. They are very stable and they do not fuse with other chromosomes. They kind of stick away from each other. and they are extremely important in maintaining the structural integrity of the chromosomes, especially during cell replication, DNA replication for cell division. We will be talking about that as well when we talk about replication of DNA during S phase. And I have again talked about that before, and I'll link that video down below in description as well for those of you who would want to look at that a little bit more. So here you have again an image of an electron micrograph of chromosome. You see the chromosome arms here, and you see the point of constriction, the primary constriction, which is your centromere. And you see these banding patterns that's formed when you stain them with certain basic dyes, which is why they were called chromosomes. So the two sister chromatids, these are identical. This is what you would get after replication of DNA in S phase. And so these are going to be what combine together in and stick together at that centromere. Now, chromosomes, like I said, don't have to have that centromere right in the center. They come in all different flavors. So depending on where the chromosome has the centromere, where the centromere is one way that we classify the chromosomes and name them. So metacentric chromosome is where this centromere is right at the center, like we located near the center. So both P and Q arms essentially are equal or nearly equal in length. A submetacentric, on the other hand, is where it's displaced towards one end, creating a long and a short arm. An acrocentric is where you have one of the centromere very, very close to one end. So you end up with a long arm and a little knob or a satellite on the other end. And finally, a telocentric is a chromosome where there's essentially no short arm. There's only two arms. It's right at the very, very, very end or near the end of your chromosome. Another set of terms that we want to know about chromosomes are the difference between homologous and non-homologous chromosomes. Homologs or homologous chromosomes are pairs of chromosomes, are members of a chromosome pair. So the two chromosome ones, your or the two copies of chromosome 1, the one from your mom and the dad, or the two copies of chromosome 4, those are going to be your homologous chromosomes. Now, chromosomes that contain different genes, right? So the two homologs or the homologous chromosomes are going to contain the same exact genes in the same exact sequence. There may be different flavors of the genes, right? So there may be different alleles, one for blue eyes and one for brown eyes, or whatever the case may be, but they are going to be the same genes. On the other hand, in your non-homologous chromosomes, you're going to contain different genes that do not pair during meiosis, um, and that are not related to each other in most cases. So a chromosome 1 and a chromosome 2 chromosome um, is going to be non-homologous chromosomes. So right here you can see four chromosome pairs. These two, are homologs. These two are homologs, each one in each pair. These are your homologous chromosomes. But when you're looking at two different chromosomes, completely with the complete DNA pattern is different. It's 3 versus 4. It's non-homologous chromosomes. The homologs are what are going to pair during meiosis in our cells for cell division. Finally, now let's kind of look at the relationship between the chromosomes and the chromatids. So when you have your unreplicated typical chromosomes inside your nucleus, um, when they are going through cell cycle and they start to go into S phase or DNA synthesis phase, the first thing they will do is they would replicate the DNA to produce a sister chromatid. So each chromosome will replicate to make an exact copy of itself, which is going to be your two sister chromatids. These two sister chromatids are physically connected at the centromere. That's how we know that they are combined together. They're the same. and they're produced by replication of a single DNA molecule. So they are completely identical, those two sister chromatids. Non-sister chromatids, on the other hand, so these are going to come from the same chromosome. Non-sister chromatids are going to be from two separate homologous chromosomes altogether. So two sister chromatids from chromosome 2 joined together versus two sister chromatids of chromosome 3 joined together. Those are going to be non-sister chromatids. They will contain same gene loci in same order, but could be different alleles. So non-sister chromatids um, are going to be, here is an example. So you have your homologous chromosomes. You have two chromosomes, one from your mom and one from your dad of chromosome 2, let's say. Each of those replicate to get you now the sister chromatids on each one joined together. And so these two that are joined together at the centromere are sister chromatids. However, over here, when you're looking at these other two that are, so this is from your mom, let's say this is from your dad, these two are again sister chromatids. But this sister chromatid from mom versus this sister chromatid from dad is going to be your non-sister chromatids because they are the same genes in the same order, but they're coming from different a different homologous chromosome pair. On the other hand, a homologous chromosome versus a non-homologous chromosome is going to be two completely different chromosomes altogether. So chromosome 2 versus 3, that's non-homologous chromosomes. But non-homologous, non-sister chromatids are just same genes, same everything, but they're coming from the two different chromosomes, one from your mom and one from your dad. So that's the difference between non-sister chromatid versus sister chromatid and homologous chromosomes versus non-homologous chromosome. Now, DNA, when it is getting copied in a typical way, is going to change the way the chromatin is condensed. Sometimes because you need to have more DNA open and the DNA be more open, the chromatin would need to relax and become more available. And other times you need to have it more condensed because it's not needed anymore and it's going to, again, be condensed even more with the help of your nucleosomes and with the help of your histone complexes. Now, that remodeling to reposition those DNA based on the need, whether it is to open it or to close it, happens through specific proteins and ATP-dependent chromatin remodeling complex. So these proteins come together, the remodeling proteins come together to interact with the histone DNA complex, and they allow it to open up or to wrap up even more. This is an ATP-dependent process, and it moves the DNA along to either unravel it or to tightly bind it to those histone complexes with the help of these chromatin remodeling proteins. So we talked about how you have those heterochromatin regions, which are really tightly compacted. In that heterochromatin region, you will have, again, different proteins that are coming in to modify those histones and to keep those proteins extremely tightly compacted or to keep them more open in space. So in the heterochromatin region, one of the ways that you keep that chromatin densely compacted is through post-translational modifications of those histone tails. Remember, we talked about how the H3 had that little tail that could kind of act as a clip and hold the DNA down. So similarly, you can have histone complexes coming together, bound together with the H1 and the H3 and the tails, the histone tails that are going to hold the DNA down because they're getting modified on top to maintain that interaction. They won't do that all normally, but they will do it in this case. So the heterochromatin-specific histone tail modifications, they're usually methylations actually that kind of maintain that structure. They will come in and they will clip that DNA together, kind of hold it down into that heterochromatin structure. And then in the euchromatin region, you will not have those tails holding the DNA and will allow that DNA to be a little bit more open. Many times there are barrier DNA sequences that are present to maintain those euchromatin regions that should always be open or that should be more open than the heterochromatin region. So these heterochromatin specific tail modifications will continue to happen through the heterochromatin specific proteins, these histone modifying enzymes, until they reach the barrier sequences. When they reach the barrier sequences, that complex falls off and cannot continue further into the euchromatin region, allowing the euchromatin region to remain open and to remain more available for transcription needs. So, like I said, these histone tail modifications is what controls which area of DNA is going to be more compacted versus other, and thus which area of DNA is going to be more active or inactive. Like I said, many of these modifications, and at the end of the day, all the histones have tails. Some are more available for modifications and others are not. Histone H3 is known for a lot of modifications. So these are what I'm showing you here in this little sequence. Is that N-terminal tail with all the modifications that can be done on there. And you can see there are a lot of methylations, a lot of acetylations, a couple of phosphorylations. Now, certain amino acid sequences, right, can get either acetylated or methylated, right? So this arginine just getting methylated, but then some of them are getting methylated or acetylated. And that or is important because when it's methylated, it's more compacted. When it's acetylated, it's more open. So those combinations determine which areas are going to be more open and which areas are going to be more compacted. In histone H3, the other thing that can happen is that some of them are going to have multiple methylations, not just a single methylation. They can have a trimethyl complex on that particular amino acid. So here in the heterochromatin formation, so here in this amino acid, the lysine at position 9, you can have 3 methylations on that lysine, and that's going to cause heterochromatin formation and gene silencing in that region. Um, and remember, these are the histones, not the DNA that's getting methylated. It is the histone that has that DNA wrapped around it. So this is like the one layer of regulation that you have to control which areas of your DNA are going to get transcribed and which areas are going to be silenced. Now, on the other hand, again, the same area, but if you had lysine 4 at lysine at position 4 methylated, trimethylated, but lysine at position 9 acetylated, it's going to allow for a more open conformation and allow for gene expression to happen on the DNA that's underneath, that's wrapped around it. So very, very small changes can control which area is going to be open and closed for activation or transcription in this case. So a lot of the times, if you can notice on here, it's the lysines that are getting methylated specifically, or arginines that are getting methylated specifically in that space. Finally, we're going to talk about what a karyotype is and how do changes in those karyotype lead to functional problems and phenotypical problems in an organism. So karyotype is essentially the complete set of chromosomes that are possessed by an organism. It is a picture, the typical picture that you see is from a metaphase chromosome that are lined up in descending order. So when you're looking at these, that first chromosome should be the longest one. second should be smaller, three, so on and so forth. So here is an example of a human karyotype. So you have your 1 through chromosome 22, the somatic chromosomes that are all lined up by descending order, and then your last one is your sex chromosome. So this particular one is from a female, it's XX. The second one is from, so this is your 23rd pair, and then down here is a one from a boy, so it's XY. Um, these are prepared from actively dividing cells. So many times we use either white blood cells or bone marrow cells from your meristematic tissues of plants, for example. So just actively dividing areas of your body, whether it's human, animal, or plant. And then in that particular section, we chemically preserve the cells, kind of hold them so that they can maintain that mitotic image. We stain and photograph them after essentially treating them with chemicals like Taxol or colchicine, which is going to prevent them from entering, like finishing up that cell division. They've made their chromosomes and now they're kind of stuck there. And then you photograph, enlarge that photograph and organize them according to the descending order. This resulting image at the end of the day is called a karyotype. Okay. Um, in the karyogram, as I just mentioned, I talked about the somatic chromosomes and the sex chromosomes. While these two types of chromosomes are important because the sex chromosomes are what are going to determine the sex of that organism or that plant or an animal that may or may not look alike, right? You have the combination of those sex chromosomes that are associated with male versus female. So in mammals, fruit flies, and some flowering plants, you have XX as female, just like in humans, and XY as male. In others, where, for example, in birds, moths, and butterflies, you have the opposite, where two Zs are considered male and a ZW is a female. Um, in a homogametic XX females, ZZ male chromosomes can pair together and segregate, same as autosomal chromosomes, which are your somatic, essentially, chromosomes, during meiosis one. But in a heterogametic chromosome pair, where it's either the XY males or ZW females, the chromosomes pair, but they are not going to be uh, going through any of the crossing over or anything like that during meiosis. Now, autosomes are, on the other hand, all the other chromosomes paired that are of the same length, centromere location, banding pattern. So they're essentially your homologous chromosome pairs. Those are your autosomes. And with the sex chromosomes, that pair is maybe not the same looking. So just to give kind of an idea on different types of chromosomal abnormalities that happen or that can happen during cell division, during their duplication or replication process. We're going to go over these a lot more in detail in the next lecture. But for now, just going through the different things that can happen. The first thing is obviously you can have deletion of a small piece of the chromosome, whether it is one base pair or multiple base pairs, but you can have a deletion leading to the shortening of one chromosome significantly or just a little bit. You can also have duplication of regions within the chromosome, especially when you have genes that have a lot of repetitive sequences. That's where you have more likelihood of this happening. And in that case, you would have one homolog being longer than the other because it is being duplicated in certain regions. Another possibility or another abnormality that can happen is inversion of a particular area of the chromosome during recombination. If something goes wrong, you can have inversion of an area of your homolog in that process. All of these can have deleterious effect if they are in important genes or in an important way where they are now leading to either duplication of a particular gene that was important for its the control of which, the amount of which was important for the proper function of the body. Another thing that can happen is that you can have insertions or translocations from different chromosomes altogether, so non-homologous chromosomes. So all these, the deletion, duplication, and inversion that we talked about were in the two homologs or homologous pairs of chromosomes. But you can also have insertions and translocations between two different chromosomes and non-homologous chromosomes. And that obviously is more cause of concern in many cases because in that case, you are many times removing the regulatory sequences of the genes in question by moving the DNA away of the genes away from their original location. So some of the things that can happen when that happens, one of them is, obviously when you either are missing a chromosome or you are duplicating a chromosome. So any type of changes, big changes in chromosome number are called aneuploidy. A monosomy is where you lose one of the pairs of the homologous chromosomes. So looking at the karyotype, it's going to be 2n minus 1. Um, an example of that is Turner syndrome, which is XO. So you only have one X chromosome. There's nothing else for the other one. No X, no Y. And another one is a presence of three homologs. So an addition of a chromosome, entire chromosome, instead of the normal typical 2n DNA content. And that's called trisomy, 2n plus 1. And the most famous of those is obviously trisomy 21, which is known to cause Down syndrome. Another thing that can happen is polyploidy. A polyploidy is basically changes in entire set of chromosomes. So it's not tiny changes, it's full changes. So you have duplication of the entire set of chromosome leading to 3n or 4n, a tetraploid cell or an octaploid cell. So now you have multiple copies of DNA beyond the typical diploid. Now, many plants and some animals are known to be either polyploid or haploid. So polyploids, when you have a polyploid organism, are not signified typically with an N like usual. Instead, we use X. So typically when we think of a diploid organism, we say 2N DNA content, where N is basically the amount of DNA in the cell. But in the case of a tetraploid, you would say 4X because it has um, four copies of the DNA, not just the two copy of the DNA. N is the number of chromosome in the gametes. So for a diploid organism, N is equal to the number of chromosomes that are present. But if you have a tetraploid organism, N is going to equal two copies of each of the chromosomes because it is four copies total. These type of changes can lead to stable or sterile progeny. If you have even number of copies, it can be stable and it can be many times divided just like normal, and they can create more progeny without a problem. But um, if they are uneven numbers, if it's like a 7n or a 3n, like bananas, for example, are 3n, those plants are typically sterile and you can only make them, you can create progeny through clippings or through uh, combining them with a diploid or a even number organism to get more progeny out of that. Many times the polyploidy leads to healthier, larger crops or healthier organisms. And so they're used a lot in agriculture for food, for just, making crops that are more resistant, that produce more. For example, the wild strawberries that are diploid are very small, right? But the octoploid, the ones that you see in the market, are much more plump and robust and large. many crops for wheat and for for general use that we have are hexaploid and octaploid. So here are some examples of plants that we use in our diet, in our life, in a typical day, and that are multiploid or that are polyploid. Bananas, watermelon, a lot of these seedless plants are triploid, so these have three copies, hence they're sterile and need other ways to be propagated in long term. Another thing that you can have is monoploidy, just like you can have polyploidy, where you have many, many copies of the genes or chromosomes. You can also have monoploidy, where you have only one set of your entire genome. There's no backup or there's no second copy. Um, Organisms that only have one set of chromosomes are usually inviable for most of the species, but hymenopteras like bees, wasps, ants, their males are monoploid and they develop from just unfertilized eggs. So if the egg is fertilized, you get a female because you get a diploid organism, but otherwise you just have a haploid male. um, so male drones, for example, in this case, this is a haploid diploid sex determination system rather than a, those XY or ZW system. So in this case, a male drone is going to be haploid, so it will just have one copy of the genome, and a diploid female is going to have the two and full set of a diploid, two copies of the genome. And the sperms in this case are going to be formed just through mitosis, just typical normal mitosis, no difference. But eggs would still need to go through meiosis so that they can create haploid gametes that can then combine with the sperms to create diploid females again. The last interesting concept with the chromosomes that we'll talk about, and I love these, is the endoreduplication. This happens in very, very specialized cases in special types of tissues that are specific for genome amplification. This happens in many, many plant cells and very, very specialized animal cells. It never affects the germline or gametes because it is happening in usually like the salivary glands of flies or some specific types of tissue only. It's not all over the cells or all over the organism. In this case, cells undergo multiple rounds of DNA synthesis, so they duplicate their DNA multiple times. So you're essentially getting that polyploid cell without making a polyploid organism. Um, they don't go through mitosis, no cytokinesis. They just keep duplicating the DNA multiple times. So you have these chromatids all joined together in these chromosomes, making these really thick chromatids. These are usually associated with cells that are extremely metabolically active and that are doing a lot of work. And they are great research models for genetics because you can see them under the microscope so well, and you can identify the banding pattern and you can dissect them and look at them at find what's wrong with them, what happens when you remove a part of it or not part of it. Here is an example of these type of chromosomes, chromatids in the salivary glands of Drosophila melanogaster and fruit flies. And like I said, these are called polytene chromosomes because they have multiple chromatids all joined together. And they are super cool to observe and super exciting to work with when for geneticists that are interested in studying these bands and genes and chromosomes. So why would it be that we would want to have organisms that are polyploid or aneuploid? Well, that just, they just arose for different reasons around because of whatever environmental changes were happening. But basically, when we look at those gametes of organisms, you will see that there are a lot more polyploid organisms present and they are tolerated a lot better than something that's aneuploid. So remember, in polyploid, you just have the entire set of chromosomes. Everything is just duplicated or multiplied multiple times. In aneuploid, you have specific portions that are messed up, right? That just um, or it's just one chromosome that is changed, one chromosome that is lost or added. So that aneuploid, you would think, hey, there are more changes if you have the entire set duplicated, but somehow that's tolerated a lot better than if you have a single chromosome that is duplicated or deleted. And the biggest reason for that is the way our body is set up, our cells are set up. We have several levels of gene regulation, several levels of regulation controlling what proteins are getting made, when they're getting made, how much of them are getting made. And so having an entire set of instructions still allows for a lot of those regulatory pathways to be maintained, to be kept intact, so that those changes can be accommodated for. On the other hand, having just one chromosome deleted or added breaks down some of those instructions in a way that it cannot function, it cannot compensate quite as easily as if just everything was up and down. And so that leads to, because it's just a particular set of area that is duplicated or deleted, it leads to the inability to compensate for these changes and leads to more abnormal or lethal phenotype results. So in summary, for this particular section, we we talked about chromosomes today, and we talked about how they are very complex and dynamic structures that are composed of DNA and proteins in the form of chromatin. The degree of compaction is related to the needs of the cell, the stages of cell cycle, and it varies between heterochromatin, that is tightly, densely compacted chromosomes, versus euchromatin regions that are more open and available for transcription. Chromosomes can be easily distinguished cytogenetically or cytologically based on their length, where the centromere location is, the banding patterns that are present when stained, as well as with sequence-specific probes to identify specific regions of the chromosomes. Homologous chromosomes are basically chromosomes that pair together that have the same series of genes along their length, not necessarily the same flavor of those genes, same alleles, but they are going to be exactly the same sequence of genes. They, when duplicated, have sister chromatids that initially contain the same alleles as well. Several types of defects can happen in chromosomes just because of errors in replication during DNA replication, and that can affect both the cell function as well as long-term evolution of that organism. Aneuploidy results anytime you have addition or subtraction of a whole chromosome. So errors in chromosomes are just small sections getting deleted or inserted or translocated. Aneuploidy is when the entire chromosome is getting deleted or added in. And those can lead to usually very abnormal or deleterious effects to the cell, many times leading to lethal effects in the organism. Polyploidy, on the other hand, is when it is not just a chromosome, not just a section, but the entire set of your genome duplicated or added on. You have one less or one more. And this is extremely important in development of crops, in development of organisms, and evolution of organisms. You can have even numbered multiple sets of chromosomes that can be stably inherited, or you can have sterile organisms because of odd sets of chromosomes. However, they can still be have progeny using other methods. This is very common in agricultural crops. And then finally, we talked about endopolyploidy, which is how you get those polyteen chromosomes, where specific tissues in your organism are polyploid. And this is observed in many, many, many species. Um, we showed you an example of salivary polyteen chromosomes in Drosophila. This can also happen in other diploid organisms as well, and we'll talk about some examples of those later. Both aneuploidy as well as any kind of structural defect in sections of chromosomes because of insertions, deletions, translocations will affect overall gene balance and would lead to deleterious response, could lead to genetic disorders of various types, and we'll see examples of them all throughout the semester. Many of those smaller changes within the chromosomes are what are known to cause cancers of various types, and whole aneuploidy changes are what cause many of the genetic disorders that we see nowadays as well. So next week, we are going to be looking at mitosis and meiosis specifically, and we are going to be talking about how they affect these specific types, how they can cause these specific types of changes, the aberrations that we talked about today. So how they can lead to these chromosomal abnormalities and how they can lead to aneuploidy changes inside the cells, leading to those genetic disorders. So I'll talk with you then. Take care. Bye bye.